that make sense?
Yes.
Okay, that is something, and they will teach you in the programs that you go into. They're going to teach you how to do the calculations so that when you're looking at the lab work, you're going to know what to understand about that lab work. Okay, so if my goal is to get you to understand what they mean by osmol. Okay, so if it breaks apart, how many does it break into? Does it break into 2? Does it break into 3? Okay, and then they'll use that for testing purposes. And we usually are told something in the millis. Because, for example, I couldn't have 1 entire osmol of sodium because that would mean the entire weight of it, which is almost 23 grams. Think about 23 grams of, well, first of all, I don't know if y'all know about sodium in itself, just by itself. It has to be kept in a liquid environment in the lab or it explodes. Does everybody remember that from part 1, hopefully? It's really cool, okay? It has to be kept under heavy oil because if it gets exposed to even 1 tiny, tiny drop of water, which is in the air around us everywhere, it blows up. It's actually used as a bomb in World War II. They could literally just drop it into like a pond or something and kapow, blow up. So cool. Okay, so think about that. We have this very explosive metal, all right, and then you got chlorine, a gas, deadly. Y'all know that, right? Okay, it's used in World War II gas chambers, okay? But you take the 2, put them together, they precipitate to these little crystals we eat. So, don't you love chemistry? I know, chemistry's cool.
When you explain it like that, you make it sound fun. Yeah.
Oh, sure. Yeah, because you know that anything on the left is a metal, anything on the right, we're running into mostly gases. A lot of the stuff on the left you can blow up, okay? A lot of stuff on the right, you know, can be deadly, all right? You got those little transition metals in between that are like, oh, okay, I know you need me, come find me. All right, and then you got all that, you know, lanthanide and all of the nuclear stuff. That's like, okay, yeah. I mean, chemistry is pretty cool when you think about it. So, when you look at information, salinity lets us learn about the sodium chloride. The reason that we tend to kind of focus on sodium chloride is because if you think back to how muscle contracts, how nerves pass along their conduction, we are looking for sodiums, potassiums, calciums, chlorines, okay? And sodium is very important in the body. Potassiums, calciums. Now, a hematocrit, that is telling us information about how many red blood cells. So in that separation that can occur, that bottom layer that had those red blood cells, that would let us know what is that amount. And then, 37, 48, 45, 52, they're looking for how much of that sample was taken is the red blood cells. So roughly about half should be those packed red blood cells. Hemoglobin. What's that? Alex?
Part that carries the oxygen.
Oxygen. It's hemoglobin.
It's a guess, but isn't it like a protein and shit?
It's protein. Okay. Now, I know, poor Emily. She's over here like right in the way of all my hands swinging it every day. But don't worry, it won't hurt. Okay, so, all right, think about this. Okay, I mentioned the red blood cell. Not gonna keep its nucleus, but that red blood cell has to mature. From the time that red blood cell gets formed until it becomes mature is about 5 to 7 days. In that 5 to 7 days, it has all of its cell parts: nucleus, endoplasmic reticulum, ribosomes. And guess what it spends that 5 to 7 days making? My protein hemoglobin. Because we need it to do one thing: carry my oxygen. So when it's mature, it spits out all those organelles, and it's full of those hemoglobin proteins so that they can attach the oxygen and transport it. And they live about 120 days. Red blood cell count, we should have a certain amount that shows up in that layer. Platelet count, we should have a certain amount. White blood count, we should have a certain amount. When they look at this type of information, usually you may have that blood drawn under a normal checkup, and they come back and give a lot of this information to you on the lab reports, okay? If a person is sick, they may draw the blood, so maybe they can see, well, if the white blood cell count is high, then they know the body's fighting something, all right? Like, for example, allergies that are coming up. Anybody in here suffer from allergies? All right, y'all can already hear it from me, all right? And that would mean probably my eosinophils and some other ones are increasing in number, trying to help me fight the allergies, all right? But we know it's going to happen, all right? So they look to, I mean, they have so much now that they can get from this information. It's amazing. I can remember when I was growing up and we had to go like to the doctor for checkups, whatever the case might be. The one thing I used to hate, I used to always hate going because I knew they were going to take that little needle thing, poke my finger, get that little tube, and let the blood drop in it, and somebody was going to walk off. Okay? Yeah, I hated that thing, you know? And so that's just something I remember from growing up. So as we look at those components, as we have that blood plasma, as we have the buffy coat, as we have the red blood cell area, the red blood cell part. But remember, as it's traveling through the vessels, it's not separated. It's not separated into these components. Now, the matrix, okay, the blood plasma, that is our matrix, that connective tissue, because that's what blood is. It is actually a tissue. In plasma, look what's present: water, proteins, nutrients, electrolytes, nitrogenous waste, hormones, and gases. Plasma's busy. Now, when we look at blood and we get it, and if we take out any clots or solids that might be in the blood, that remaining fluid is termed serum. You may see sera, okay? The sera contained blank, maybe a way that you see it. It's almost the same as plasma without a clotting protein. That clotting protein that we're going to find is called fibrinogen. This is a very important molecule that's going to be found in our blood. Fibrinogen. What does it make you think about? Fibers. What are the 3 fibers of the blood? Of our body, I should say.
Collagen.
Collagen, elastic, reticular. Collagen is the biggest and the toughest. Elastic, like the name implies, it's kind of stretchy. Reticular is the smallest and creates weaving that can occur. But now, wait a minute. Can I have fibers as they exist, as the collagen, as the elastic, as the reticular? Can I have those moving around in my blood vessels as a fiber? No. So what I have instead is this. And note the term that's being given to it. Clotting protein. When do I need a clot to form in my body? When I need healing to take place. Any other time, it creates what we know as a blood clot, which can definitely be harmful, right? Mm-hmm. So, fibrinogen, let's go back for a second to my puppy bites, all right? I need that to heal. I need that to heal. Okay? And it's formed the clot. Why? It gives rise to the fibers. It gives rise to the fibers. So at the time of the wound opening, because fibrinogen is present in the blood, it can rush to the site that's available and begin to create a web. And that web allows platelets to get stuck, and I create the clot. This is happening in the body 24/7 because we have minor breaks throughout the body all the time. Little blood vessels burst and repair throughout the body all the time. We're not even aware of it. We're aware of what we see, but we're not aware of the other stuff.
That's what happened to me because I thought I was getting a staph, and I kept rubbing my eye because it hurt, and now I got a blood clot in my eye.
Like the little red spot? Yeah, yeah. But it'll heal. I mean, did you go to the doctor? Did they?
It just happened like yesterday. I didn't even know I had it. My mom seen it.